Okay, welcome to the laboratory orientation of blood and bone marrow. Okay, this organ in focus is the spongy bone, and this actually the part where this is your bone marrow, showing you the different stages of formed elements such as your red blood cells, your white blood cells, and your platelets. Now, the spherical spaces found scattered here at the bone marrow are your adipocytes. Adipocytes are also known as your fat cells. This is the bone marrow. Encircled, the cell pointed right now is the megakaryocyte. This is a stage of platelet development or thrombopoiesis wherein megakaryocyte, the cytoplasm of which no fragmentation of the cytoplasm of the cell being pointed right now would result to platelets. Okay, so the most common questions asked is identify the cell pointed. This is your megakaryocyte. Structure where this is found is the bone marrow, and fate of this cell pointed is through fragmentation of the megakaryocyte will lead to the formation of platelets. Okay, the cell pointed in yellow arrow is your megakaryocyte, the fragmentation of which would lead to formation of the platelets. Platelets are found in the blood vessel or in our blood circulation. Let's go now to the different white blood cells. The white blood cells is categorized as being granulocyte and agranulocyte. Showing you here are the granulocyte white blood cells. Number one is the neutrophil. It is described as having a multi-lobe nucleus with pale red and blue cytoplasmic granules. It only has one nucleus and it is multi-lobe, usually 3 to 5 lobes that are present in the neutrophils. This one is eosinophil, exhibited by having a bilobe nucleus with red cytoplasmic granules. This one is the basophil. Basophil is or has bilobe nucleus with purplish black cytoplasmic granules. Granulocytes is divided into two. We have lymphocyte and monocyte. Lymphocyte is exhibited by having a large spherical nucleus with thin rim of pale blue cytoplasm. The ratio of the nucleus to cytoplasm is the nucleus occupying most of the cell. Monocyte, on the other hand, is a agranulocyte exhibited by having a kidney bean-shaped nucleus with abundance of pale blue cytoplasm. Okay, red blood cells, its lifespan is 120 days. They are described as non-nucleated with biconcave disc. The average diameter of red blood cell is 7.5 micrometer, and the main function of which is to transport oxygen from the lungs to the tissues and carbon dioxide from the tissues to the lungs. Neutrophils is the most abundant white blood cell found in our body. They're also known as your polymorphonuclear white blood cells or segmenters. The normal value of neutrophil is 55 to 65%, and the significance of which, if there is an increase in neutrophil count, is you are having a bacterial infection. The lobes here is usually from 3 to 5 lobes, but the number of nucleus, as I mentioned a while ago, is 1. Next is the lymphocyte. This is the second most abundant white blood cell found in the circulation. It is characterized by having a large nucleus that occupies most of its cytoplasm. It is increased in viral and chronic infection. Eosinophil, on the other hand, is described as having a bilobe nucleus with eosinophilic cytoplasm. It is increased in helminthic and parasitic infection. Not only that, it is also increased in allergic reactions as well. Basophil is described as having a large bilobe nucleus with purplish black, dark blue granules. If you notice in this picture, this is the basophil. It almost always the nucleus is not visible anymore but it is a bilobe nucleus. It is characterized by having an abundance of granules all over the cell. It is increased in hay fever, bronchial asthma, hypersensitivity reaction, and allergic rhinitis or allergic dermatitis. They are very specific for IgE antibodies, although they are also increased in allergic reaction between eosinophil and basophil, but this is specific only for an IgE receptor. Monocyte is the largest cell in the circulation. It is exhibited by having a kidney-shaped or bean-shaped nucleus. Its cytoplasm is more abundant. It has a grayish blue tint. They stay in the circulation for an average of 72 days, hours before they are deposited into tissue and they are converted to macrophages. The main function of the monocyte is that they are involved in phagocytosis and they defend the invading microorganism. And platelets. They are described as being a biconcave disc with 2 to 3 micrometer in diameter. They also are non-nucleated. They are in clusters or group. The normal value of platelets is 150,000 to 300,000 per cubic millimeter. Fragmentation of the cytoplasm from megakaryocyte would lead to the development of platelets. Megakaryocytes are only found in the bone marrow, whereas platelets are found in the peripheral circulation. Thank you for listening.